In this topic, we're going to look at genetic fingerprinting. So we're going to look at the stages of making a genetic fingerprint, interpreting the genetic fingerprints, and then the uses of genetic fingerprinting. Genetic fingerprinting is a diagnostic tool used widely in forensic science. The technique uses the fact that any organism's genome contains many repetitive, non-coding bases of DNA. The process is based on the fact that 95% of human DNA does not code for any characteristic. These non-coding DNA bases are known as introns and contain repetitive sequences of DNA called core sequences. Now, for every individual, the number and length of the core sequences has a unique pattern. They are different in all individuals except for identical twins. Even the probability of twins having exactly the same core sequence, length and sequence, is very small. These core sequences are more similar the more closely related any two individuals are. Right, let's have a look at the stages of making a genetic fingerprint. There are 5 stages. You've got extraction, digestion, separation, hybridization, and development. We're going to look at extraction first. Even the smallest sample of animal tissue, for example a drop of blood or a hair root, is enough to give a genetic fingerprint. Whatever the sample, the first stage is to extract the DNA by separating it from the rest of the cell. You then amplify this DNA using the polymerase chain reaction, which we've discussed in the last topic. Then the next stage is digestion. The DNA is cut into fragments using restriction endonucleases. The endonucleases are chosen for their ability to cut close, but not within the group of core sequences. Then there's separation. The different sized fragments are separated by gel electrophoresis. The fragments are separated by size under the influence of an electrical voltage. The gel is then immersed into alkali to separate the double bonds into single ones. The single strands are then transferred onto a nylon membrane by a technique called southern blotting, as you can see on the right there in this diagram. Okay, hybridization. Radioactive or fluorescent DNA probes are now used to bind onto the core sequences. The probes have base sequences that are complementary to the core sequences and bind to them under specific conditions such as temperature and pH. The process is carried out with different probes, each of which binds with a different core sequence. Then there's development. Finally, X-ray film is put over the nylon membrane. The film is exposed by radiation from the radioactive probes. If you're using fluorescent probes, the positions are located visually. At these points correspond to the position of the DNA fragments as they've been separated during electrophoresis. So you get a series of bars, as you can see on the left there. Right, let's interpret the results. If you have a look at these genetic fingerprints, you can use this to identify which child comes from the mum and dad. So you can see which bands correspond to one another. So have a look at this genetic fingerprint to identify which children are related to the mum and dad. You can see that daughter 1 and son 1 are related to the mum and dad because their bands correspond to either the mum's band or the dad's band. What about daughter 2? Has she got the same father as daughter 1 or a different father? She's got a different father because her bands do not correspond to this dad's bands. And then son 2 must be adopted because neither of his genetic fingerprints, the bands from them, correspond to the mum or dad. So when we're interpreting the results, the genetic or DNA fingerprints from two samples are visually checked. If there appears to be a match, the patterns of the bars of each fragment is passed through an automated scanning machine, which calculates the length of the DNA fragments from the bands. The more the two patterns match, the greater the probability that the two sets of DNA have come from the same person. So here you can see that the blood on the defendant's clothes match the victim's blood. Okay, let's have a look at some uses of DNA fingerprinting. Now, it's used extensively in forensic science to indicate whether or not an individual is connected with the crime. So looking at this, which suspect was at the crime scene? Must be suspect 3. And then it can also be used to help resolve questions of paternity, since every individual inherits half the genetic material from the mother and half from the father. So looking at this diagram here, which father is the child's father? Must be father 2. And then finally, genetic variability. So it's useful in determining the genetic variability within a population. The more closely two individuals are related, the more closely their genetic fingerprints will resemble one another. And that concludes our lesson. The end.